It's my distinct pleasure to have the opportunity to introduce Professor Claire Roberts. Professor Claire Roberts is an inaugural NHMRC Leadership Fellow and Matthew Flinders Professor of Flinders University, where she leads the Pregnancy Health and Beyond, which I'm guessing you pronounce PHaB?
We do, with the PHaB Lab.
With the PHaB Lab, which I have to say is the cleverest name of lab so far. I mean, I think it's NOAA's SMAC Lab is close, but PHaB Lab, I think, actually takes, it's hard to, okay. She is also an adjunct professor at the University of Adelaide. Professor Roberts is the immediate past president of the International Federation of Placenta Associations. She's an internationally recognized authority and thought leader in placental research. She's worked on placenta from 8 different mammalian species, but human placentas, we are told, are her favorite, if you choose favorites. Claire is scientific director of the Adelaide Scope and Stop Pregnancy cohorts and has developed patented algorithms for use as screening tools to predict which women are at high risk of developing a major pregnancy complication. Professor Roberts recently completed the NIH-NICHD Human Placental Project study to profile patient placenta and maternal blood across gestation using next-generation sequencing technologies. She has a particular interest in modifiable risk factors, including maternal diet and micronutrient status and other lifestyle factors for poor health in pregnancy and beyond. She studies genetic factors, including fetal sex, that contribute to placental function and pregnancy outcomes at the population, cohort, and placental transcriptome level. She's a perfect speaker for us in terms of our transdisciplinary Center for Evolution and Medicine Studies. The title of her talk today is What Can Placenta Tell Us About Health in Pregnancy and Beyond? And with that, we will finally surrender the floor to Professor Roberts. So thank you, Professor Roberts.
Thank you very much. So thank you for that introduction. I should start with my disclosures. Ken mentioned my patent. I'm a mother of 2 young women and a grandmother of 4 children, which in the context of being a pregnancy researcher, I think is important. I live in Adelaide in South Australia, and you're going to say, where is that? And so here's Australia, here's the US, so we come a long way to speak. I am here for a couple of other things as well, so I'm quite excited to be here. And you can see Australia is actually a big country. It does fit inside the US, but it's almost as big. And really, I like to think it's actually the top of the world, right? It's all about where you're looking from, right? So where is Adelaide? Adelaide is the capital of South Australia, which is here. You may have heard of Sydney. The Olympics were there in 2000. You might have heard of Brisbane or Melbourne. You probably haven't heard of Adelaide. But you see all the black dots on that little map, that's where all the people are. So we essentially hug the coast and mainly the cities. And we only have about 26 million people altogether. And most of the land in here, there's supposedly green here, but there really isn't much green there. It's really desert right in the middle. It's tropical up north, and it's temperate down south. We don't have any snow here in Adelaide, and I don't think you do actually in Phoenix, right? So I think our climate is fairly similar. Maybe it's a bit colder here in the winter. So that's where we're from. So I like to start with this because in pregnancy health is dictated largely by what happens in the placenta as it develops in the first trimester of pregnancy. So in these photos, you can see there's a 6-week human conceptus, 8 weeks, 10 weeks, and 12 weeks. And you can see in this early part of development, it's really the growth of the placenta that is dominating, and its differentiation is a dominating force on future pregnancy outcome. So I like to think of the placenta as controlled cancer. It grows like a tumour. It becomes hypoxic, has to undergo angiogenesis like a tumour. but also it's highly invasive. If we look here, this is the placenta, this is the maternal tissue, here's the decidua, which is the name for the pregnant endometrium or lining of the uterus, here's the myometrium, the muscle layer. And all these cells are extravillous cytotrophoblast. Extravillous because they don't belong to the villi, the chorionic villi. And what they do is, particularly in early pregnancy, they invade into the decidua. They colonise the uterine spiral arterioles. They considerably transform its structure such that the vessels become non-responsive to vasoactive molecules. And this process takes many weeks, and paradoxically, early in pregnancy, right up to 10 weeks, there's actually no maternal blood flow into the placenta, into this intervillous space, until about 10 weeks, and that establishment of maternal blood flow takes 3 or 4 weeks to occur to its maximum. So in early pregnancy, the placenta and obviously the embryo is developing in a hypoxic environment, and that's essential for pregnancy success. But you can appreciate why I say it's like a tumour. It's a tumour and it's essentially metastatic, if you like. Fortunately, choriocarcinoma or placental cancer is quite rare. So just to show you the hypoxia, so this is days of gestation on the x-axis, and people have actually measured oxygen tension in the decidua, so the uterine tissue, and in the placenta prior to that onset of flow. And you can see here is the uterine tissue or decidua, and really its oxygen level is about what you'd expect for other tissues. Here's the placenta tissue. So right at the beginning, oxygen tension is very low, and it gradually increases, and it plateaus off to about the tension that you would see in any arteriole in the body. And people have also used ultrasound, Doppler ultrasound, to profile blood flow, and you can see in this very early gestation placenta there is no maternal blood in the placenta. So here again is a first trimester placental villus sample. This was taken by a Dutch collaborator of mine. Here's the sample that they get just with a needle, and you can see there's a stem villus here, and these fluffy things are chorionic villi through which all exchange will occur. You can see even though this is 11 weeks, some of them have fetal capillary development, others do not. So that process of angiogenesis takes place over many weeks as the placenta grows and develops. And all the while it's delivering oxygen and nutrients to the fetus. And at term, this is what it looks like. This IVS is intervillous space. Here are the chorionic villi, labelled with cytokeratin to show you the syncytia trophoblast that covers the entire surface. So that's the surface for exchange. This intervillous space is where maternal blood flows. So you get a real interdigitation or dipping into maternal blood of chorionic villus tissue, which of course is fetal tissue. So just as every baby is unique and looks different, so too does every placenta. They're genetically identical to the fetus as a product of conception. You can see they all look different. The insertion of the umbilical cord is in different parts, and they're not all round as you might expect, so they're very variable. And some of them have what we call a filamentous insertion, that is, the cord enters through the fetal membranes. And if you see here, these are umbilical vessels entering into the placenta, and it's a wonder that this baby didn't have a catastrophic bleed when it was delivered. So you can tell a lot about the health of that pregnancy just by looking at the placenta. And of course, sometimes they look different just because the research assistant who collected it did something different to it. So I said we've only got 26 million people in Australia, and there are about 300,000 births a year. This is for 2020, where our birth rate went down, likely due to COVID, mainly because we weren't taking migrants for that year. And about 30% of these pregnancies have a late gestation complication. Not all life-threatening, but 30%. So 1 in 3 women will have a complication. And if I think you looked at American data, it would be very similar. Some women have more than one complication at a time. Probably, you would say 5 to 6% of pregnancies, a complication may be life-threatening to either the mother or her fetus. And pregnancy complications are a continuum. So they have a lot of overlapping etiology. So we talk about pregnancy being 40 weeks from the last menstrual cycle, and all sorts of things can happen from miscarriage, which typically happens before 20 weeks. Fetal death after 20 weeks is stillbirth. You can have fetal growth restriction, preeclampsia, which by definition occurs after 20 weeks, preterm birth, which by definition occurs after 20 weeks. Term is 37 weeks, but we talk about gestation as 40 weeks. Pardon me. Gestational diabetes can be diagnosed at any time during pregnancy. Typically in Australia, we diagnose it between 24 and 28 weeks, but that doesn't mean that a woman might not be diabetic prior to that diagnosis. But some of these complications occur in the same woman in the same pregnancy. They have overlapping etiology. The placenta is associated with all of them. So what is this thing, the placenta? I'm assuming you don't know much about the placenta. I looked at your website and I didn't see it mentioned. So I'm assuming you don't know anything about it, so forgive me if I say things that you already know. So obviously the placenta is the site of exchange between maternal and fetal circulations. The blood circulations do not mix. And the placenta delivers oxygen, micronutrients, macronutrients, drugs, viruses, basically anything that the woman might even rub on her skin, sniff, inhale, or whatever, is potentially transferred to the fetus. And then the fetus, of course, gives up its waste products to the placenta where they're directed to the maternal circulation for elimination. The placenta makes many hormones, both steroid and peptide hormones, that control maternal adaptations to pregnancy and growth and development of the fetus, but also for its own function. So the health of the placenta is very important to the health of the mother and, of course, to the health of the fetus. But the health of the mother and things to which she's exposed strongly impact the health of the placenta. So whatever the mother is doing can potentially affect health of the placenta and hence health of her fetus. And not just things she might be exposed to, but her own health, her pre-existing conditions, her obesity if she has that, her nutrition, etc. But also the father has a part, obviously, contributing 1 copy of every gene to the conceptus, so to placenta and fetus. And it has been shown that factors such as paternal obesity can contribute to pregnancy complications such as miscarriage and preeclampsia, and that is mediated by effects on sperm health. But also then there's a suite of imprinted genes which are expressed from just 1 copy, either paternal or maternal genome, that are expressed in the placenta and by the fetus that govern its function. Paternally expressed genes tend to enhance growth. Maternally expressed genes tend to inhibit growth. And of course, these genes are also part of the demand signaling system that the fetus confers to the placenta and hence to the mother. So just a small point on the imprinted genes. Imprinted gene expression can associate with factors in the maternal physiology that change during pregnancy, including her blood pressure. This paper is out of Cambridge in the UK, and they showed a number of imprinted genes, particularly the fetal allele, that were associated with maternal blood pressure. So this is a fetal allele being expressed in the placenta associated with the blood pressure in the mother during pregnancy. And only one of these you can see is actually also the maternal allele has an effect. So the placenta, I mentioned, secretes abundant hormones and peptides, growth factors, etc., and they promote maternal adaptations to pregnancy, including plasma volume expansion. Maternal plasma volume goes up by about 1 litre. Inflammation, thrombosis, so women who are pregnant are more likely to suffer blood clots than when they're not pregnant. These hormones induce insulin resistance and pancreatic beta cell expansion, nutrient partitioning, weight gain in the mother, immune tolerance, that is tolerance of foreign antigens expressed by the placenta and fetus, and they also alter every other organ in the mother to enhance their function. And of course, in the fetus, they promote growth and organ and tissue differentiation. Here are some of the hormones that are made by the placenta that enter the maternal circulation. And you can see there are neuropeptides, pituitary-like hormones, adipokines, growth factors, steroid hormones, and I can't read that from there. Oh, adrenal-like peptides. So all of these are made by the placenta, and they govern and orchestrate the adaptations of the mother. And then, of course, her own genome governs how she responds to those. So there's a real crosstalk between the fetus, the placenta, and the mother to enhance or not pregnancy success. So here's just a couple of plots showing you those changes. Here are the hemodynamic factors, and you can see even prior to 10 weeks, a lot of the hemodynamics are changing rapidly, for example, like blood volume. So they're changing even as early as before the woman even knows she's pregnant, but in that first trimester when she doesn't even have a bump to show that she is pregnant. No wonder women, you may have known women who are pregnant and in first trimester, they got nothing to show for it except they're exhausted. And it's no wonder, they're still busy doing a heck of a lot of things. I think I'll skip the rest of that. So these major complications of pregnancy that we're interested in are preeclampsia, preterm birth, fetal growth restriction, gestational diabetes mellitus, and stillbirth. They're all associated with how the placenta develops. I mentioned about 30% of Australian pregnancies, and I think that would be similar in the US, are affected by these, and these have consequences not only for the mother and her fetus during pregnancy and gestation, but also for her health beyond, and I hope that I'll give you evidence for that. So we've recruited a number of pregnancy cohorts in Australia, so in Adelaide, and we recruited from a hospital that serves a community of low socioeconomic status, so very disadvantaged women. This one is nearly 1,200 women, and this cohort we called SCOPE. It's part of an international cohort of 5,628 women in 6 centres in 4 countries. And you can see here are the other centres, so over 5,600 women. And we were intending to develop screening tools that could predict in early first pregnancy. So all these women are nulliparous, so they hadn't ever had a baby before. So we have no idea what their risk is. If you have no pregnancy history, we don't know what your risk is. So we wanted to find ways of screening women to find out which women were at risk. So we have actually published a lot on this cohort. And all these factors that are in purple, obviously, you can't do anything about. So your chronic diseases that you may also already suffer from, your ethnicity, your family history, whether you have a male or a female fetus, all of these things we can't do anything about, but they can affect outcome. And then there's a whole lot of modifiable factors. Things like maternal asthma, smoking, alcohol, use of illicit drugs, folate. We have a very big interest in folate and the use of folic acid, which I'm not going to talk about today, but we had a conversation over breakfast that it's very important. So there are lots of different factors that affect health in pregnancy. It's no wonder that we haven't really nailed what is that factor that's really important in preeclampsia, because there are lots of different factors and they interact differently in different women who are partnered with different men. because paternity also influences these pregnancy complications. We have another cohort that we recruited in Adelaide at the same hospital 10 years later. And the idea was we had developed these algorithms, we wanted to validate them, which we have done. I'm glad to say. We've collected a huge amount of information from these women. So clinical information, obviously, psychosocial, their education background, their diet. We've taken blood samples, urine samples, and we have developed large clinical databases and biobanks from these. So we're still continuing to use these. So health in pregnancy, as I said, there's an awful lot of different factors that govern the health of this placenta and the baby. Obviously, they include things like genetics and epigenetics. So epigenetics is affected by some modifiable factors like nutrition, obviously ethnicity, but also low socioeconomic status is associated with increased risk for all pregnancy complications. And that doesn't just speak to privation, it also speaks to education. I heard today some women here in the US, the biggest economy in the world, don't get any prenatal care visits, which I find shocking, because we know that more prenatal care increases and improves outcomes. So all of these different factors interact with genetics, alter epigenetic state in the placenta that contribute to outcome. But things like maternal age, older women, women who are obese, women who don't have sufficient exercise, they're all at increased risk. So you can appreciate there are a lot of different things interacting for health. And they affect how well that placenta makes its hormones. So how it does orchestrate maternal adaptations. And they affect every single thing you can think of. There are a whole lot of other things, of course, that I haven't written here that can be altered in the placenta in different individual women, partnered with different individual men for pregnancy success. This is one of my daughters, Kelly, and one of my granddaughters, Emma. And she was a young, slim woman when she got pregnant for the first time. She was 29, so not old. She was slim, so one wouldn't have thought she was at risk, but she developed gestational diabetes in this pregnancy. Why did she do that? We think she had a good lifestyle, upbringing, education, all of that. Was she just unlucky, or was it something to do with her partner? Well, no, there's no family history. No family history in his family of diabetes, right? But we know, of course, he's given one copy of every gene to that fetus and that placenta. So he is likely to have some sort of an impact. I'm not apportioning any blame here. But also, the quality of his semen. So there is abundant research showing that, for example, cytokines in seminal plasma govern how well the woman adapts to and becomes tolerant of her partner's antigens. So you think about it, the antigens on sperm, the antigens on placenta and the fetus are foreign to the mother. She has to become immune tolerant to his antigens. So we and others have published that if you have a short period of sexual cohabitation with your partner or a short period without the use of barrier contraception, you're more likely to get preeclampsia and you're more likely to have a small for gestational age baby. So having a period of sexual cohabitation of certainly at least 3 months, preferably at least 6 months prior to conception, improves your pregnancy outcome. So there's a good reason for monogamy, actually. What else? So this is Popper. So Popper, of course, is my partner. That's Malcolm. So, of course, he's Kelly's dad. So he contributed some genetics along the way. And he also contributed to these factors that we would have hoped would have improved or reduced her risk of having something like gestational diabetes. And then there's grandma. And I have found since I've become a grandma, grandmas are really important because grandmas do contribute part of their genetics, but also they contribute a whole lot more. So you think about it, when a woman is pregnant, Everything she eats, I've told you, everything she eats or drinks or smokes, inhales, rubs on her skin potentially can be transferred to that fetus by the placenta. And here's that fetus. So here's mum consuming all of this. It will govern her health of her fetus, but you think about it, the gametes that are developing inside that fetus for that fetus's children in the future are also being influenced by what she's eating, drinking, etc. And grandmas are important again because, I'm just talking myself up here, right? Because all the mitochondria you inherit come from your mother, and therefore they come from your grandmother as well. So things that you did when you were pregnant are going to affect mitochondrial energetics, or energetics in every cell in the body in your children and then in your grandchildren. So we can't think about pregnancy in isolation. We have to think about the bigger picture, transgenerational, etc. So this paper is looking at the role of mitochondria in health, but thinking about mitochondria as having a completely different genome to the nuclear genome. So we've talked about mum, dad, papa, grandma, all contributing to genetics, and now we're thinking about mitochondria, which have come, from a long way back, all contributing to different factors. So here's mitochondrial copy number here on the y-axis, and here's the trophectoderm, which will be the trophoblast, which is the business cell of the placenta. And you can see that mitochondrial copy number goes up very early, much earlier than that in the somatic tissues in the fetus. And it goes up to a much higher number. So the placenta is incredibly biosynthetic and very bioenergetic. It is a misnomer. People talk about a similar sort of plot for methylation and demethylation and remethylation. Actually, in the trophectoderm, remethylation happens much, much later than this would suggest. So let's take a step back to where it all began. What does a placental biologist know about fertility? Well, I'm not an expert. I have published a couple of papers on it, and even on the use of a growth factor to enhance embryo development. But this is probably more like the level of the sophistication of my knowledge, right? But we have published in that big scope cohort of over 5,600 women in 4 countries about time to conception and their diet. So time to conception obviously is the time that you're trying to get pregnant. We asked women how long they were trying to conceive, and you can see that this was a relatively young cohort, first time pregnant. So nearly 40% of them actually conceived within 1 month, so in the first cycle. 8% were deemed to be infertile. That is, that they took longer than 12 months to conceive, and some of those women required some sort of assisted reproduction to become pregnant. And the upshot is that with the lowest fruit intake were more likely to take longer and much more likely to be deemed infertile than those who ate more fruit. I'll show you a plot of that in a moment. And similarly, or conversely, for fast food, those women who ate a lot of fast food, over 4 serves a week, for some people that isn't a lot, but I think it's a lot, that increased their risk substantially of being infertile. So that's doing something to the gametes and to the ability to fertilise. So this plot just shows that. You can see we compared women who had 3 or more serves of fruit a day to different groups, and those who had only 1 to 3 serves of fruit a month took a lot longer to get pregnant, and a lot more of them were infertile. Here's the fast food data again, showing if you eat 4 serves a week or more, you're more likely to be infertile. And with a poor quality diet usually comes poor metabolic health. Now, women are not usually assessed for metabolic syndrome when they're pregnant, but for our research we did, and we did this at 15 weeks gestation. And again, in over 5,500 women, we showed that 12.4% had metabolic syndrome at 15 weeks gestation. So they probably actually entered pregnancy already with metabolic syndrome. So metabolic syndrome is, have I got that? No, I haven't, sorry. I've obviously left that slide out. So we used International Diabetes Federation criteria, so it includes waist circumference, circulating lipids, cholesterol, total cholesterol, HDL, LDL, and also blood pressure and glucose. And those women who had metabolic syndrome were much more likely to need assisted pregnancy, but also we've been doing some work looking at, we did a systematic review looking at the infertile women and what their future health might be. So it turns out that in a large number of women, so if it's difficult to become pregnant, it's also associated with future risk for cardiovascular disease. So this forest plot shows the mean difference in blood pressure, BMI. So women who have a history of needing fertility treatment have higher BMI later. They have higher cholesterol later and higher triglycerides later, but similar, and higher LDL cholesterol later, but similar HDL later. So the fact that they had a history of infertility predicts future cardiometabolic disease. And then if we think about metabolic syndrome in pregnancy, it's also associated with pregnancy outcome. So again, we use IDF criteria, which I mentioned before. And you can see here that women who had metabolic syndrome, so people will say, oh, it's just because of they're obese. But if we look at BMI as a continuum here, this is probability of getting gestational diabetes. It's going up, but it's very modest. But if those women also had metabolic syndrome at 15 weeks gestation, their risk increased nearly fourfold. So having the combination of metabolic syndrome and obesity substantially increased their risk for gestational diabetes. And conversely, having that combination reduces their probability that they'll have an uncomplicated pregnancy. So metabolic syndrome affects your ability to become pregnant and your ability to have a healthy pregnancy. So gestation is changing. The length of gestation is changing. So this is South Australian data, but I have actually taken the next slide out, which is this plot, but it's just showing that over time, women are delivering earlier, and a lot of that is about obstetrician intervention. So we're having more inductions of labour, more caesarean sections that are planned early. And some of that is because women are being detected who actually need that. Some of it is for convenience or for money. But what are the other trends? So this is over 500,000 women in South Australia over time. And you can see it starts in 1986 and this is up to 2014. And you can see over time, women are getting older. And that trend is the same in America, right? Women are having their babies later, mostly because of careers, trying to establish their careers prior to having a baby. Women are also more likely to be overweight or obese now than they were 30 years ago. You just need to walk down the street, and I'm old enough to have walked down the street 30 years ago, or been at school. When I was at school, there weren't any overweight and obese kids in my year, right? And now you go to a high school and they're nearly all overweight, right? So I mentioned 25 to 30% of pregnancies are affected by these. They affect maternal and fetal health perinatally, but also they increase risk for developing non-communicable diseases later. And did you actually know, because we hear in Australia an awful lot about breast cancer, of what a big killer of women it is, but actually 3 times as many women die of heart disease as die of breast cancer. So it's actually much more important. And I don't know about America, but half of the people who suffer a heart attack, they die before they get to hospital. And a lot of those people are going to be women who were thought to be at low risk because they were premenopausal, but they're not. But if we knew about their pregnancy complications, we might have been able to monitor them better and identify them earlier, such that when they had that chest pain, they didn't just think it was indigestion. Or that chest pain actually is not the typical in women, it's not the typical symptom that men get. Women have some very vague symptoms when they're having heart attacks, and so they get palmed off a lot, so they don't get treated early enough. So often they don't even get to hospital. I'm going to focus a bit on long-term consequences of gestational diabetes. All pregnancy complications are associated with increased risk for chronic disease. Gestational diabetes I want to focus on because you can see how it's increasing over time. This is from 2000 to 2018 in Australia. I believe the numbers are similar in the US and in other developed countries. So something is happening. It's not about obesity, because over that time where there's an exponential increase in the incidence of gestational diabetes, obesity is about the same. So it's not about obesity, it's about something else. I had a conversation at breakfast about folic acid. This is when we introduced folic acid fortification. Now, I don't have time today to talk about it, but it may be harmful in some women. Okay, so, and this is South Australian data over time, and again, it's also increasing exponentially. So if we use our now current criteria for gestational diabetes, this is a scope cohort. You can see the incidence was low. It was actually 5% of the scope cohort in Adelaide, and it's gone up to 15% in our STOP study, which was 10 years later. So what are these women at risk of? Well, we've done a number of systematic reviews and shown that women who have a history of gestational diabetes are at increased risk. So there's a lot of papers included in this. There were, I'm pointing at the wrong thing, 140 of these 12,000 odd papers were eligible, 93 were included in the meta-analysis, and that's pretty hard to read, so I'm going to focus on the important part. And this is about 50,000 women in this part, and you can see that they are, and this is between 1 year and I think it goes up to 25 years postpartum, their blood pressure is higher. That doesn't look like much, but that actually is clinically significant because they don't have heart disease, but their blood pressure is higher, and blood pressure, as it goes up, it damages your blood vessels, particularly your endothelium. So these women, because they have a history of gestational diabetes, have higher blood pressure, and in fact they have higher systolic and diastolic blood pressures. Their BMI is greater, their total cholesterol is up, their LDL is up, their triglycerides are up, their fasting glucose and insulin up, and their HDL, supposedly protective, is lower. So that history of gestational diabetes identifies women who have cardiometabolic risk factors that likely will result in their needing a hospital visit at the least. What about their children? So we looked at the children and their risk, and here there are 59 eligible papers, 25 included in meta-analysis, and you can see that these children, so between 1 year and 15 years after their birth, they have higher blood pressure than those who were not exposed to gestational diabetes in utero. They have a higher BMI, so their BMI Z-score is higher, which translates to over 1 kilo per metre squared BMI. These are young children. So what are they going to be like in 10 years' time, in 20 years' time, in 30 years' time? they also have higher fasting glucose. That's nearly 0.5 millimole of glucose. If you consider that the reference range is 3 to 5.4 millimoles per litre, having nearly 0.5 millimoles per litre higher fasting glucose in childhood is going to greatly increase their risk for type 2 diabetes. And indeed, those children who are exposed to gestational diabetes do have higher rates of obesity, etc. So it turns out the risk factors for the pregnancy complications are identical to the risk factors for ischemic heart disease. So I think in this part, I hope I've told you that pregnancy foreshadows future health for both the mother and the child, and there's abundant more data than I've had time to show you. But we really need to know, well, what are the molecular and cellular mechanisms that underpin this? And of course, we have to look to the placenta that is orchestrating pregnancy health. So I was very fortunate to get one of NIH's Human Placenta Project grants in 2016. And the idea was to do a molecular profile of the placenta and matching blood samples in women from 6 to 23 weeks gestation, and then at term in our cohorts for women that we also had blood samples for throughout pregnancy. We haven't achieved that yet. We haven't integrated all the data, but we've done RNA sequencing, microRNA sequencing, and used Illumina DNA methylation epic arrays to assess transcription and obviously methylation. We've also done microRNA sequencing in matched blood samples taken from the women at the same time of their terminations, and also the methylation in the circulating leukocytes in the mother. So this was our first paper. This is unfortunately still a preprint, but it's coming. And this is profiling early gestation RNA from 6 to 23 weeks. So you can see here this PCA plot nicely shows the gradation of gestation. So things are changing with gestation. Here you won't be able to read the gene list, but there are a whole lot of genes obviously that are differently expressed between 6 to 10 weeks, which is that pre-maternal blood flow. That's the hypoxic phase of gestation, with 11 to 23 weeks gestation. You can see we had similar numbers of males and females, not identical, but across these time points. So if we look just with this volcano plot, you can see that there are, on the left here's 6 to 10 weeks, a whole bunch of genes that are higher in the placenta at that time, and then distinct genes that are more highly expressed in the 11 to 23 weeks. So there were a total of over 1,000 genes that were differentially expressed, some up, some down. And many of them are genes that we would potentially expect to be involved. For example, like MMP12 is involved in trophoblast invasion into the maternal decidua and the blood vessels. What have we got on the other side? I can't read it. Oh, there's angiopoietin-like protein 1, and that's actually previously been associated with preeclampsia, as has TGF-beta1. So there's some interesting things that are changing in the transcription of genes that we know are abundant in the placenta and associate with pregnancy complications that are changing in that early period. So we also did a WGCNA analysis. This is Jimmy Breen, who was the bioinformatician associated with this work. And you can see with this analysis, the modules, their expression is changing across time. Here's that 10 to about 13 week period where the gene expression actually flips. So here's our presumably hypoxic period. Here's the transition while maternal blood flow is established, and here's post that about 14 weeks period when the placenta should have normal oxygen tension, or what we'd call physiological oxygen tension, which we thought was very exciting to be able to see that. And to see and get evidence that there is this transition period, it takes time, that it's not just like that suddenly you've got this great oxygen environment. And here's Melanie Smith in our group, who's been meeting with Seema and Melissa online. This is work she did on microRNAs, again at that 6 to 23 week period. And you can see also the plot shows that gene expression is changing with gestation. And she looked again at those two different time periods, 6 to 10 weeks compared to 11 to 23 weeks gestation. This is the chromosome 19 microRNA cluster. And you can see that its expression decreases after establishment of maternal blood flow. And this is the chromosome 14 microRNA cluster. And you can see that it's actually increasing in expression after the establishment of maternal blood flow. She's also looked at, do I have it? No, I don't have it. She's also looked at the microRNAs. We did microRNA sequencing in plasma, and we can also see a change after that period of the establishment of maternal blood flow. The interesting thing about that chromosome 19 cluster is it's primate specific and it's placenta specific. So if we see any of that in maternal blood, it is coming from the placenta. So in theory, it can indicate health of the placenta. At some point, we'll get to doing the integration of all our data so we can truly, I hope, develop a test where we could take blood at any time in gestation, profile it and say, that reflects the molecular profile in the placenta. That placenta and thereby that fetus are going well or they're not. We need to do something. So just to change plot again, sex differences. So sex differences start before implantation. So in humans, implantation occurs about day 5 or 6 after fertilization. And already in the pre-implantation human blastocyst up here, there are about 600 genes that are differentially expressed between male and female pre-implantation embryos. In fact, they start at 2-cell embryos when the genome is activated. Males on average grow faster than females in utero. They're actually more at risk because they're on average 100 to 120 grams heavier. That's about 4 to 5 ounces heavier than females, so 4% heavier, and that goes right across gestation. Males appear to make their placentas work much harder than the female placenta. So the ratio of newborn weight to placental weight is higher in males, so they squeeze every last drop of nutrition out of that placenta. And the faster growth of the male puts it at much greater risk of adversity in utero. So those fast-growing males are more likely to die and hence be stillborn, or more likely to be born preterm in some sort of adverse environment, such as if the mother is asthmatic and has an asthma exacerbation. Conversely, the female fetus grows to survive. So males they keep on growing. So for example, if their mother has an asthma attack, they just keep on growing, they don't stop. So they risk dying. But females, if the mother has an exacerbation of her asthma, they taper their growth. So their strategy is a strategy to survive. And if you think about that from an evolutionary point of view, how many males do we need? They just need to, well, you think about other species, right? So they grow fast because they've got to be big. Bigger babies survive after birth. Bigger babies grow to be bigger adults, so they're going to be more competitive in terms of being able to transmit their genes to the next generation. Whereas females, most of them will reproduce. So females just need to survive. So we're seeing that strategy in utero. And males, we've done some work, actually we've done a a study, a population-based study in South Australia of 550,000 births over 30 years, and shown that males are more likely to be born very early preterm than females. They're more likely to be born to a gestational diabetic pregnancy. Females are more likely to be born very preterm only if their mothers have preeclampsia or hypertension. And preeclampsia is more common in the male-bearing pregnancy at term than the female. So males are known to have more adverse outcomes. And that continues after birth. So very preterm babies, boys, are more likely to die than girls. They're more likely to suffer respiratory distress and bronchopulmonary dysplasia, which is a big factor in the survival of very preterm infants. But even the glucocorticoid that we give to the woman who comes to the labour ward in threatening to deliver early, to deliver preterm, we give her a glucocorticoid injection to try to mature the infant's organs, particularly the lungs, but all of them, so that that baby will survive after birth. But if you use betamethasone or dexamethasone, the outcome is different if you have a male or a female. And many hospitals, they just use one. Most hospitals, in fact, just use one, the one they've always used, so they don't know what it's going to do, and they don't always know that there's a male fetus, for example. So even that is affecting outcome. So just thinking about sex differences, this is Melanie's data again on microRNAs, and you can see these are the male-specific microRNA network that's differentially expressed in the placenta between 6 to 10 weeks and 11 to 23 weeks. And you can see there are sort of 4 hubs there with 4 important microRNAs that have been associated, some of which have been associated with pregnancy complications. Here's the female-specific pattern, different microRNAs. And interestingly, there's this gene at the hub, and that hub, so the target of all those microRNAs which repress expression are acting on IGF-1 receptor. And that will, if you inhibit that, you'll inhibit growth, because IGF-1 is a big growth factor in the fetus and in the placenta. So in the female, the microRNA suite is targeting anti-growth, if you like. So this is our scope data from 5,628 babies, and you can just see that at every gestation in blue, boys, this is the gestation of their birth. The boys are in blue, they're heavier than girls, so at every gestation, and girls are in red. But interestingly, the extremes are more common in boys. So we've also looked at sex differences in the placental transcriptome. This was work done by Sam Buckbury, who was a graduate student in our group some years ago. And while he was waiting for some sequencing data, he looked at some publicly available microarray data that was around at that time. And he had data for 303 placentas and found 142 genes that were differentially expressed between males and females. You can see from this Circos plot, obviously the chromosomes around the circumference here. Obviously there's some hot spots on the X and Y chromosomes, so the further out the red dot, the more significant is the gene differential expression, but also really right across the genome there are differentially expressed genes, and actually 2/3 of them were autosomal and only 1/3 were X or Y linked. So some were going up in males and some were going up in females, and these were normal singleton placentas. So nothing wrong with these pregnancies, but the male and female placentas are quite different. So that's what you can see at term. We've seen work showing early gestation differences. So the male and female placenta are completely different. This is Melanie's work on preeclampsia. It's a GSEA analysis. She found, like Melissa's group, no differences between the preeclamptic female placenta compared to an uncomplicated female placenta, only differences in the male placentas. So the preeclamptic female placenta looks very like the placenta from a female of an uncomplicated pregnancy. These are different gene pathways, of course, that are associated in boys. So there was a great suite of genes that were differentially expressed between male preeclamptic versus male uncomplicated pregnancies. So they're very different. They're in pathways we'd think about. The immune system, obviously, the immune response is very important in pregnancy complications. Inflammation is a feature of pregnancy complications, as is oxidative stress, which I haven't had time to talk about today. So I hope I've convinced you that pregnancy foreshadows future health for women and their children. We shouldn't just get stuck on one thing. I haven't been able to talk to you about micronutrients, which we're doing a lot of work on, and a whole lot of other lifestyle factors, ethnicity, etc. But so we shouldn't get stuck on any one thing. But this is my message to you. This is a quote from 1901 from the Equal Franchise Association in Australia. Australia was one of the first countries in the world to give women the vote, because these strong women were advocating. They said, women are a class that is half the world and mother to the other half. So we should be focusing on the health of women, the health of children, the health of the placenta and pregnancy outcome. It not only foreshadows health for the mother, but foreshadows the health of her children. And that means it foreshadows the health of the whole of the population. But we don't spend much money on women's health research. And we absolutely must. And everybody who's heard me today, I hope to hear that you advocate for that in the future. Thank you. And I should just thank all the people in my research group. I've mentioned Melanie, she's up here. These are my postdocs. Shalom is a statistician who developed our algorithms. Got some students along here. Here's Melissa and Emily, who've done a lot of work on metabolic syndrome and future cardiovascular risk. This is Gus Decker, who's my very long time clinical collaborator. Margaret Arstall, who's the director of cardiology at this hospital that we've recruited our cohorts from. We now run clinics for every woman who's had a pregnancy complication. She comes to the clinic, she sees a cardiac rehabilitation nurse, and she has a suite of hemodynamic and blood testing to look at her cardiometabolic risk. At 6 months postpartum, she's seen again with the same tests at 18 months, and those women are getting counselling to improve their cardiometabolic health. We're trying to start that at the new hospital that I've moved to, because that's really important. These are young women that we can prevent these chronic diseases. Thank you.
Thank you for a great talk. We have time for a couple of questions, and while people are queuing up, I'll ask one. How well understood, or what is the mechanism of that crosstalk that you talked about that differs between males and females in response to her health?
I think it's very poorly understood, and that's why we really need. I would like to see NIH investing a lot more in the Human Placenta Project. Unfortunately, they're not. So they've really suspended funding for that. But it's absolutely essential that we understand, because only when we understand and we can identify molecular markers, for example, that we can target them and actually do something about. But, obviously understanding is the first step, right? And we just don't know.
Yeah, so great talk. Really interesting. I was wondering about the gestational diabetes, and you showed that sharp rise. You also presented kind of a 3-generational model. Could it be that it's not the current obesity, because you showed that obesity rates are essentially stable in that population right now, but could it be that differences in maternal obesity or grandmaternal obesity and changes in that, 1 generation or 2 generations back?
So they're grandmas? Maybe. So a generation before that, so I would say in Australia, women started getting fatter probably in the 90s. And so that really is a generation before this population we're talking about now. And so it's quite conceivable that sort of dietary changes, people were getting more money, they were more likely to go out, get takeaway food, those sorts of things. So yes, potentially, and that could have affected the gametes. There are other things that are changing though in that time. I mentioned folic acid fortification. So we started that in 2009 in Australia. And I think I showed the plot that showed that our scope cohort was prior to folic acid food fortification. The STOP study is 10 years later, it's after that. And we're actually working now, I don't think I've got the graph with me, but we're working now on an association between folic acid and gestational diabetes, because it has gone up since then in Australia. It's not straightforward. Folate status has gone up remarkably, so that the average folate, serum folate and red cell folate in women now is absolutely through the roof and above the reference range for folate in the population. Now that's in these women we've measured, so in the cohort before and after, over 2,200 samples. But it's not as simple as that is associated with gestational diabetes. We have published before that women who had high folate status did have higher incidence of gestational diabetes. That holds true for our subsequent studies. In the STOP cohort, though, it doesn't matter if women supplement, it makes no difference. Their folate is already through the roof. What is that doing to methylation in the placenta? But it's not, and the women in those two cohorts, well, the first one, because they were a deprived cohort, they had a high rate of overweight and obesity anyway. So obesity's gone up a wee bit. But there is data out of the US, out of China and out of India that shows an association with gestational diabetes and folic acid supplementation. What we've done most recently, and we've just submitted a paper this week in fact, first of all on women who have an uncomplicated pregnancy, and show that we compared the before and after, they have higher folate status. But also they have higher, we've measured placental lactogen, placental growth hormone and prolactin, all of which govern insulin resistance, because you have to become insulin resistant when you're pregnant, and your beta cells compensate for that. So you get beta cell expansion and greater insulin secretion. Not all women compensate sufficiently, and if you don't, you get gestational diabetes. Well, the women who had higher folate status, and certainly in our second cohort, they have altered profile in early pregnancy up to 16 weeks gestation. Those hormones are different. This is in uncomplicated pregnancy, and our next manuscript is looking at pregnancy complications, particularly gestational diabetes, but their hormone profile is different. So the hormones that govern insulin resistance and beta cell expansion are different post-fortification to prior. Is it having a direct effect on the placenta? We think it is. We've done some placental explant studies where we've treated them with different doses of folic acid and measured the hormones secreted into the media, and the growth hormone variant is altered in response to high levels of folic acid. In fact, it looks like it's a bit of a U-curve. So being deficient in folate is bad news for placenta and fetal growth. And the growth hormone variant is higher. Then it goes down at sort of normal physiological levels, and then it goes up again. So we think it is having a direct effect on the placenta. The next step after that is to have a look at, does it actually affect beta cells? So we do think there's something important happening there. We also have, because we hold a lot of methylation data before and after in our cohorts in the placenta, we're looking at also, does the folate level actually increase or alter methylation? And, you know, you'd have to say it's likely, right? So we think it could be changing the expression of a suite of genes within the placenta.
One last quick question.
Oh, okay. Thank you. Great talk. Towards the beginning, you were talking about a lot of lifestyle factors like diet, smoking, exercise that affect the success of a pregnancy. If someone has a poor lifestyle, then decides they want to become pregnant, can they change their lifestyle and then have a successful pregnancy, or is it due to a lifetime of those choices?
As far as we know, I think you can change it. I mean, I think anything is modifiable, and whenever you do it, it's got whatever's is going to be better afterwards for having done it. Certainly, we've actually looked at diet prior to pregnancy and very limited things like fruit consumption, green vegetable consumption, fast food consumption, now dairy we're looking at. And a better, if you eat fruit and green veg, you're likely to have a better quality diet. And certainly it does seem to be the preconception period that's important for that. So diet in pregnancy, yes, you should have a good quality pregnancy, a good quality diet, but preconception matters. And we asked women about the 3 months prior to conception. But to me, if you improve your diet before you conceive and give it a few months of a decent diet, then I think, the odds are that it's going to be you're going to benefit from that in pregnancy. It is never too late to improve your diet and get exercise, right?
So, as much as I hate to cut the questioning short, Dr. Roberts will be here, Professor Roberts will be here longer, but I want to take the opportunity yet again to thank you for an outstanding presentation and a lecture and a seminar, and look forward to further interactions and ask the audience to give one more round of applause.
Thank you.